Hello everyone, I am going to be presenting my journal article titled De Novo Translation Initiation on Membrane-Bound Ribosomes as a Mechanism for Localization of Cytosolic Protein mRNAs to the Endoplasmic Reticulum. This is written by the four writers below the title. I'm going to be going through the background, methodology, results, and conclusion. So first, I will kind of explain the figure on the right. This just shows a translation of mRNA through cytosolic ribosomes and through membrane-bound ribosomes on the endoplasmic reticulum. On the endoplasmic reticulum, those have the signal recognition particle, as I'll be discussing in just a second. So basically the main point of this article is about mRNA localization. So this is important to control protein synthesis, and as we're learning in class, eukaryotic cells localize signal peptide encoding mRNAs to the ER by the signal recognition particle, or SRP, for translation. So while this SRP pathway is well established in mRNA and protein sorting to the ER, there is actually evidence for additional mechanisms of mRNA localizations to the ER. So this new evidence, as stated on the PowerPoint, indicates translation of cytosolic mRNA on the endoplasmic reticulum, or the ER. So the cytosolic protein encoding mRNAs have actually been identified on the ER, but because there's no actual encoded signal sequence for the localizations of these mRNA to the ER, makes this topic very controversial. So that is why these researchers decided to study this. I'm going to go through some terminology, just because I know that most of us don't know these. In vivo just means in a living organism. GAPDH is something that the paper talks about a lot, and that's just a cytosolic mRNA. And then GRP94 is a signal encoded mRNA that has association with the ER-bound ribosomes. So the experimental hypothesis, as stated, is if the endoplasmic reticulum-bound ribosomes are initiation competent in vivo and do not select RNAs based on signal, then the cytosolic mRNAs can undergo translation on these ribosomes. So this just means that they do not select for signal peptide encoding mRNAs. So if they do not select for these signal peptide encoding mRNAs, then the cytosolic protein encoding mRNAs can undergo translation on the ER-bound ribosomes. So basically this research will determine if cytosolic protein mRNAs can be endoplasmic reticulum localized. So what would allow this? So they believe that translation initiation, if both cytosolic and endoplasmic reticulum ribosomes are competent for de novo translation regardless of trafficking of encoded polypeptides. So really, this basically means that the signal recognition particle, or SRP pathway, would direct localization of signal sequence encoding mRNAs from cytosol to ER, which would be the reason for cytoplasmic protein encoding mRNAs to be able to be synthesized on both the cytoplasmic and endoplasmic reticulum ribosomes. So we all know that these cytosolic mRNAs will be mainly synthesized by by the cytosolic ribosomes, and also the same for the endoplasmic bound ribosomes will be mainly synthesizing the mRNA that has these signal encoding sequences. But this paper is just wanting to dig into how the cytosolic mRNAs are being found to be translated by these membrane-bound ribosomes. So here are a few materials and methods that this research had to do to be able to get their data. So there's cell culture, detergent extraction, RNA extraction, and PCR, protein extraction, and immunofluorescence staining and single molecule fluorescence. So this brings me into the results. So this figure basically as a whole uses the single molecule RNA fluorescence, which basically allows visualization of individual mRNA molecules visible by diffractions of limited spots. So this figure basically reveals that cytosolic protein encoding mRNAs are actually in close approximation with the endoplasmic reticulum. So in figure A, it just shows that the protein and RNA from cytosol and membrane-bound are distinguished by the immunoblotting that's taking place. So it shows that the cytosolic membrane are showing up on the two that should be, because GAPDH, as I stated, is a cytosolic mRNA, and then the same for the bottom two, showing up for the signal sequence encoding mRNAs, and there's that GRP94. In B, it's an immunostain used to detect and better distinguish the cytosolic mRNA versus the ER-localized mRNA. So that's what's being shown there. In C, we see that GAPDH and GRP94 are visualized, and there was found to be an ER protein marker in both, which is highlighted on the figure, or boxed. And then lastly, D is the immunostaining and the signal, or the single molecule RNA fluorescence that I talked about in the beginning, which shows the overlap of GAPDH mRNAs and GRP94 mRNAs, which shows evidence that these cytosolic mRNAs can be localized to the endoplasmic reticulum. So this next figure is a box whisker plot of RNA samples that were analyzed by qRT-PCR, and as you can see, basically this represents that various mRNAs, including cytosolic cytosolic mRNAs, are on the endoplasmic reticulum, is what is shown through this graph. So this is consistent with the single molecule RNA fluorescence data reported in the first figure. And so the overall point is that it demonstrates the subcellular mRNA from the cytosol is parallel to the ribosomal distributions. So this third figure shows the correlation of 18S RNA, which is a component of cytoplasmic ribosome. So it shows the correlation of 18S RNA and GAPDH mRNA on the ER membrane in different cell lines. So in figure A of this, it's a plot of the fraction of endoplasmic reticulum-bound ribosomes versus the fraction of endoplasmic reticulum-bound GAPDH mRNA for all cell types examined, and this revealed a high correlation. So from both these correlations, you conclude that the subcellular distribution of GAPDH encoding mRNAs is linked to subcellular ribosome distribution, which itself determined by the amount of endoplasmic reticulum present in a given cell. So this is another figure that really pushes their hypothesis to being correct, that these cytosolic mRNAs do associate with the membrane-bound ribosomes on the endoplasmic reticulum. In this figure, this shows mRNA from the cytosol undergoing translation on endoplasmic reticulum-bound ribosomes, and it shows that they couldn't distinguish between the scenarios where the cytosolic mRNAs are recruited to the endoplasmic reticulum at a specific stage of translation initiation or at a later stage of translation. So that's the only part of their hypothesis that they could not actually identify it being at the initiation of translation. But the data demonstrates that cytosolic mRNAs can undergo translation initiation on the ER. So they can undergo, they just don't know if that is the initial initiation of the translation of the mRNA, the cytosolic mRNA. So overall, the ribosome footprinting patterns of the cytosolic and ER-associated mRNA populations are very similar. So this all ties in to their hypothesis of further showing that the cytosolic mRNA can be in close proximity and be translated by these membrane-bound ribosomes. So, in conclusion, we all know that cellular protein synthesis is done by both cytosolic and endoplasmic reticulum-bound ribosomes. Mainly, cytosolic proteins predominantly are synthesized by cytosolic ribosomes, and as well secretory pathway proteins are synthesized on endoplasmic reticulum-bound ribosomes, because this obviously allows or makes sure that the location of these proteins are correct. So it ensures that these proteins go to the right location in the cell. But while this is true, this paper does indeed show that there are cytosolic mRNAs that are found to be represented on the endoplasmic reticulum. So these cytosolic protein encoding mRNAs were found to be broadly on the ER, but they are still there. So cytoplasmic RNAs correlated with the membrane-bound ribosomes, as talked about in the paper. And these RNA distributions could possibly mean that this mRNA localization, this cytosolic mRNA localization to the ER, could be regulated by multiple mechanisms. So this paper doesn't really dig into what those mechanisms are, but it could be further research could help figure out what these mechanisms exactly are. So in this paper, it basically shows that RNA with expected cytosolic distribution of GAPDH does actually reside in close proximity to the endoplasmic reticulum and can be translated on the endoplasmic reticulum-bound ribosomes. And also GAPDH, which again is the cytosolic mRNA, was found on the membrane, which shows a significant positive correlation with ribosomes that are membrane bound. So that is the end of my presentation, and I hope you learned a lot more about how these cytosolic mRNAs are able to be found on the membrane-bound ribosomes on the endoplasmic reticulum. Thank you.